Hello and welcome. Today, we're diving deep into the microscopic world to uncover the secrets of a truly unique organism, the Mu Phage, also known as the jumping virus. I'm so glad you're here at Cognito Key, where we unlock the secrets of science, one key concept at a time. So, let's get right into it. Okay, so picture this, a microscopic invader that's so sophisticated, it doesn't just infect a cell, it actually rewrites its very essence. I mean, what if a virus could literally cut and paste itself directly into its victim's DNA, becoming a permanent part of its genetic code? Well, that's not science fiction, not at all. Let me introduce you to the Mu Phage. Now, it's a type of bacteriophage, which is just a virus that preys on bacteria. But here's what makes it so special. It has this dual identity. On one hand, it's a virus, right? Its mission is to replicate. But it's also something called a transposon, basically a jumping gene that can literally move around and insert itself into the host's genome. So how in the world does it pull this off? Well, to understand that, we've got to look at its design. And really, you should think of it less like a simple organism and more like a high-tech molecular machine built for one single purpose: invasion. So let's check out the blueprints. First off, just to give you a sense of scale, the head of this machine is only 54 nanometers across. I mean, that is incredibly tiny. Its main job is to act like a little vault, protecting the phage's precious genetic material. But what's even more amazing is how this head is actually built. See, it's not one solid piece. No, it's constructed from exactly 152 individual protein subunits. They're called capsomers, and they all fit together perfectly, forming this really tough protective shield. And then you've got its injection system, the phage's tail. This thing is 183 nanometers long, and it's this incredible contractile marvel. It basically works like a powerful syringe designed for one thing: to punch through that bacterial cell wall. So let's just quickly recap that three-part structure. You've got the head, holding the DNA blueprint, then the neck, which connects everything, and finally the tail, which even has these little fibers that act like landing gear, ready to lock onto its next victim. All right, now let's peek inside that head. The DNA in there? It's basically the instruction manual for the entire takeover. Think of it as a genetic toolkit with 55 different genes, and each one is a specialized tool designed to hijack its host. Okay, we're not going to go through all 55, don't worry, but let's meet the star players. First up, the A gene. This is the master key. It codes for the enzyme that lets the phage do its whole cut and paste trick. Then there's the C gene, which is like the stealth mode switch, deciding when to lay low and when to attack. The mom gene, this one is pure genius. It creates a chemical disguise, modifying the viral DNA to hide it from the host's defense systems. And finally, you have the LIA gene. That's the demolition charge for the grand finale. So the machine is built, the toolkit is ready, it's time for action. Let's watch how this all comes together during the invasion itself. It's a meticulously planned, step-by-step takeover of the host cell. It all starts with the breach. First, those 6 tail fibers touch down and lock on. That's the trigger. The tail contracts like a powerful spring, driving a needle-like core right through the cell wall. And just like that, bam, the viral DNA is injected inside. Then, using that A gene enzyme we talked about, it just seamlessly weaves itself right into the host's chromosome. Next up, the factory takeover. At this point, the phage makes a critical decision. Does it stay dormant, or does it go active? If it goes active, it hijacks the cell's entire replication machinery, forcing it to pump out around 100 new copies of the viral genome. And this is where that brilliant mom gene comes in, quickly camouflaging every single new copy so the host's defenses can't find them. And now for the final act, the great escape. The hijacked cell is now nothing more than an assembly line, building all the phage parts: heads, tails, fibers. Incredibly, these parts just self-assemble into new, complete virus particles. Each new head gets packed with a fresh copy of the viral DNA. And then, on command from the lysine gene, the cell literally bursts open, releasing a new army of about 100 phages, all ready to start the cycle all over again. So yeah, it's a fascinating process, even if it is a bit brutal. But the big question is, why does this one specific phage matter so much to us? I mean, what can we actually learn from its unique strategy? Well, What really makes the Mu Phage so important is that dual nature we talked about. Because it's so good at inserting genes randomly and efficiently, scientists have actually harnessed it. They use it as a powerful tool for genetic engineering and for studying how genes work. It's like a perfect natural-born geneticist. So it's not just some curious virus. It's a remarkable example of evolutionary efficiency that we can learn from and even put to work. You know, the story of the Mu Phage really shows us that the microscopic world is just filled with these incredibly complex and elegant machines. It really makes you wonder, doesn't it? What other marvels of microscopic engineering are out there, just waiting for us to find them? Hey, if you enjoyed unlocking this little piece of knowledge with us, please subscribe to Cognito Key. And of course, don't forget to like and share this explainer with anyone else you think might find it interesting. Thanks so much for joining us, and we'll see you next time.